welcome for the presentation, and I'm happy to be here. A very warm welcome. So today I'm going to present on the third HPTLC online user meeting in HPTLC and HPTLC-MS, an essential tool in the assessment and botanical dietary supplement. This is Aboli Girme from Herbal, India. Thank you so much for being here, and I'm very happy to share some of my experience and some of the research in this very prestigious event organized by ANCROM and HPTLC Association. So let's start for some of the research and latest requirement for the HPTLC for in the light of botanical dietary supplement. So what are the assessment criteria of botanicals and botanical dietary supplements? Some of the challenges may I list out with the help of definitely diversifications of multiple molecular classes of botanicals present. There are some of the challenges we do face as a researcher and as a commercial R&D from botanical authentication. Then there will be geographical and seasonal variation, the chemical diversification, obviously multiple classes present, standardization, batch analysis, and stability profile, along with the current changing regulatory requirements. So the question is how the very good chromatographic tool like HPTLC could be beneficial in all this. So let's dive in some of the very interesting history and evolutionary benefits of HPTLC development and how it has helped in the botanicals. So this is some of the data we have collected, but definitely it's an interesting how we have come up from this linomat and application, and now we are at the at where there is a rapid application available. Then there is reproducibility, high traceability, and we are these HPTLC tools has tremendously helped us into the not only the method development, method validation, now evaluation, which is one of the major criteria as a very confirmatory testing for the multi-chemicals or the chemical markers. So how the HPTLC investigations can help us into the these botanicals analysis and investigations and assessment. So when we talk about HPTLC UV or the qualitative unit, it's definitely help us in one of the major criteria, which is a botanical authentication. The HPTLC DS technique has thoroughly helped us for the standardization or the quality control unit into the chemical assessment. When we talk about a novel tool, HPTLC-MS, which has gained popularity recently, which could be very helpful to when it comes to have the thoroughly chemistry and the conformational studies of this multi-chemical marker-based botanicals. So these HPTLC analysis definitely an imperative quality control characterization when it comes to not only authentication but also the adulteration problems in the industry, and definitely into the research. So these are some of the analytical guidelines which can be used when from the beginners to the researcher labs or the industry people, which are very thorough and very detailed guidelines can be very helpful when it comes to botanical analysis by HPTLC, which includes the different pharmacopeial guidelines. Today's focus of presentation will be how these three different techniques of HPTLC can be very beneficially used from the botanicals. First of all, as identification tools in the botanical or the botanicals product, which could be anything from extract to food supplement to botanical supplement or traditional medicine. Along with it's a very popular quantitation and standardization tool for quality control and identification purpose. So HPTLC-MS is a tool which can be used for the rapid confirmation along with its isolation and identification of compound in this botany. So when it comes to HPTLC analysis, a very simple HPTLC guide could be helpful for all the users, which can be well documented, and which I will share a stepwise which can be implemented. So first step is very important is sample identification, which has been talked through and discussed very much, is identification of sample, which plant or which botanical it needs to be analyzed. So authentication is must. So authentication is very must along with it maintaining herbariums with small notes and very much details, how much details we can collect. So basically investigation applies to different botanical part like flowers, fruits, leaves, stems, and this has to be identified into dry, semi-dry and fresh conditions along with segregations and cleaning of plant material, which are very essential. The next step will be sample extraction. Before starting the analysis, the sample should be extracted thoroughly, but how the sample should be extracted, whether your samples are botanical, which is a plant material, or extract based or product or formulation based. The nature of sample and particle size of the sample are also essential when it comes to HPTLC analysis. Along with that, in some of the non-essential parts like chlorophyll, fatty acids can be removed with the help of sample pretreatment, which could be an essential step if anyone wants to include into protocol. When it comes to sample preparation, these are some of the important pointers which we need to pay attention are the temperature conditions at which we are preparing the sample, the extraction techniques, the filtration techniques. So these are some of the examples when it comes to sample pre-treatment or sample extraction protocol. For example, chlorophyll removal, where you can utilize a non-polar solvent, and then the sample could be used for further extraction or analysis part, or the sample preparation method, in which the cleaning of botanical parts to sample extraction, then filtration, along with that the particle size reduction or the concentration could be the steps which can incorporate wherever is essential. The next step will be separation of targeted analytes. So targeted analytes are the very keyword for any analyst, where there is need to know identification of which chemical or bioactive markers are needs to be analyzed. So definitely the classification needs to be done, whether it's from single chemical classes or multiple chemical classes. This can help our method development thoroughly, and which can be further the method development approach can be further compound oriented or pattern oriented. When the compound oriented approach is very much feasible when we know which kind of compounds or marker present in the particular botanical. It could be a single marker approach or multi-compound approach. When or similarly pattern oriented approach can be utilized when there are novel or unknown compounds present into particular botanicals, and it can be very useful when it compared to the reference botanical or reference material for the authentication. So during this separation of targeted analytes, different criteria plays a major role. For example, derivatization optimization. HPTLC's technique very much dependent on how we derivatize the HPTLC plate. So this is some of the examples where you can see how the different reagents plays crucial part, or identification or optimization of derivatization plays crucial part when it comes to HPTLC, along with mobile phase optimization. So different mobile phase and mobile phase optimization plays also a crucial part when there are multi-compound approach and you have to separate a botanical matrices. So this is also example of saturation time, how the time is affected. So these are some of the pointers when it should be addressed during the development phase and could be beneficial for developing a robust phase. So step 4 is a combined approach for the HPTLC analysis. We highly recommend when there is a HPTLC analysis is a very versatile tool when it comes to botanical. So definitely it is essential to combine these tools with combinational approach for qualitative, quantitative as well as confirmatory analysis. For example, you can combine the HPTLC-DS with HPTLC-MS confirmation or HPTLC-UV fingerprinting with MS or MSMS confirmation. Also, the HPTLC is a very versatile tool. The observation and findings can be combined with the LC-based PD or DED tools. So these combinational approach give a robust analysis and the findings for the botanicals. So how these HPTLC techniques or how the combinational approach could be taken when it comes to botanicals and botanical supplement. HPTLC UV or the qualitative analysis can help us into botanical authentications, and these are some of the application in which we have used the botanical authentication or the ID testing for the some of the prominent botanicals, which are also botanical products or the dietary supplement. Uh, for example, identification of flower pods, then phytogeographical studies. Phytogeographical studies are very essential and crucial parts when it comes to optimization of supply chain. Also, quality control of supply chain. Then plant material and batch analysis. These are very much applications where HPTLC, UV, or the HPTLC qualitative fingerprinting can be utilized. The HPTLC-based analytical fingerprinting and method validation definitely are the one of the versatile tools, very much comparable to other chromatographic techniques. Will not go into detail, but time has proven and many studies have shown it's equivalent standardization techniques, and it can be very beneficial when it comes to multi-class compounds analysis. So HPTLC-DS or the scanner detector can be used in different aspects when it comes to botanical analysis. So it includes the simultaneous analysis of multi-class compounds along with the identification of compounds, quantification of compounds, even the strategizing analytical investigation towards the bioactive markers and quantifying bioactive marker from batch to batch. And definitely HPTLC-DS is very much comparable tools when it comes to adulteration studies, and it increases the regulatory compliance of many botanicals and botanical. HPTLC-MS is one of the novel tools and currently getting really popular into botanical industry because of its capability of isolating and reconfirming the compounds and the bands present into different botanicals. So it has helped in different research and different research areas where you can isolate and differentiate the compounds between different species. Along with it has shown the confirmatory criteria also when it comes to botanical and botanical matrices. So these are some of the HPT which contains all of this information and in-depth protocol if anyone wants to go through that. So we'll have look at how we can optimize a sample preparation protocol for HPTLC analysis. Uh, everyone has worked on different botanicals, but with our experience, we have developed some of the optimized protocol which we use from different multiple chemical classes present in botanicals. So these protocols, or the process flow may contain the sample preparation in the form of fine powder, which is required extraction of by the solvent with the help of sonicator, or which require and inclusion of coarse powder, which may require the extraction due to the particle size or the quantity. When it comes to extraction, definitely choice of extraction is very essential for as per the targeted analytes or as per the project requirement. So if the targeted analytes are from some of these classes like carotenoids, flavonoids, terpenoids, these are some of the extraction solvent we use for the HPTLC analysis. These are basically alcohol or water-based solvent which are capable of extracting these classes of compounds. So another major factor we need to pay attention when it comes to sample preparation, that is your temperature conditions. At what temperature the sample is getting extracted. So these are some of the pointers or information which can be utilized when it comes to extraction or sample preparation for maintaining the temperature. So the samples with carotenoids or flavonoids may require a little bit lesser temperature compared to other samples, but definitely up to 60 to 80 degree are the major portion where we can keep the temperature when it comes to extraction. So further, these are some of the steps which can be utilized for the final sample preparation, in which centrifugation is a essential criteria where you can reduce the chances of the particle clean and particle-free sample solution, and which are very essential. Filtration also is one of the very major technique, and point 22 is choice of filter which can be used for HPTLC, and further it can be taken to the application. So how this optimized sample preparation can help us, and how the HPTLC analysis can further take place. So this is one of the prominent work we'd like to discuss is presenting ashwagandha or Vidania somnifera, one of the very fascinating botanical and prominent botanical currently in different global scenario. So the aim of the study was the quality control study of the bacteria classes, which represents both vidanocytes and vidanolites in the roots of Vidania somnifera. So in this HPTLC method, we could develop and separate the 7 vidanolites and vidanocytes for the quality control of the roots in Vidania somnifera. The targeted compound category was lactone and its glycoside. So the separation of 7 prominent marker was achieved, and it can be a useful method which you can see have a very less matrices effect along with a prominent separation of the 7 markers. The same HPTLC method we utilize for the extension of this study, which can be part of differentiate the other plant parts from the Vidania somnifera roots. Vidania somnifera roots are very prominent traditional medicine, and it has the similar phytochemical classes which are similar to other plant parts of Vidania somnifera. So same HPTLC method was applied for some of the other parts which are generally known as aerial parts, which contain leaves, stems, fruit calyx. So along with vidanocytes and vidanolites, in this method we have used a campherol-based glucosides, which can be prominently seen as from RF.21 at 366 nanometer after derivatization. And this can be seen into the different samples of leaves, aerial parts, fruits, and fruit calyx. This method has been validated and standardized for the same for the analysis of camphoral leukocyte along with vidanoside and vidanolide. So this analysis also helped to differentiate between other Vidania species, which is present in the latter tracks. So one of the track is 37 Vidania coagulans, along with the other foreign species, which has been reported for the adulteration of Vidania somnifera. The second example we'd like to discuss is about Tinospora species. The aim of this HPTLC method development and study was to distinguish between the two closely related Tinospora species. As you can see on the screen, these Tinospora crispa and Tinospora cordifolia, both species are very closely related morphologically. So aim of the study was to differentiate between these two species by its HPTLC profiling. So we found there are two prominent bands which are present into Tinospora crispa track number 1 to 5, which are not present in Tinospora cordifolia. Though along with other differentiating bands, these two bands found in crispa could lead us to the investigation that with the help of this profile, there is totally these two species which are morphologically very similar can be distinguished. So second part of this HPTLC investigation led us to isolation with the help of HPTLC-MS for these compounds. So we identify and we name this band as TSR1 and TSR2. So this is the protocol we developed for this study, in which first we develop the HPTLC UV profile, which showed a very good separation of compounds and band. Then confirming this band throughout the different accession or the geographical sample, we found that these two compounds are present only in Tinospora crispa samples. We generated HPTLC-MS profile of that. After that, the isolation and identification took place, and with the help of those compounds, the HPTLC analysis was performed for the CRISPR as well as the cordifolia species. So as you can see, there is a compound 1, TSR1, which is at 0.15 RF, and the compound 2, TSR2 as 0.33 RF. After HPTLC-MS, these two compounds could be further identified as borapitoside, which are very characteristic deodorant adherpent for the Tinospora crispa. So these are the structures of these two compounds we could identify with the help of MS. Further, these compounds isolated and identified with the help of HPTLC, PrEP-HPLC, and column chromatography. Similarly, the same set of method we applied for the phytogeographical and quality control studies for the authentication of Tinospora cordifolia. So here we can see how the combinational approach can work. The same set of method with the help of the isolated compounds TCR1 and 2, which are borapitosides, along with the Tinospora cordifolia compounds tinosporazide and iptosin. These two combined approach of these four compounds can be helpful for phytogeographical study and the quality control. As the Tinospora cordifolia or guduchi is a very prominent Ayurvedic medicine, but it's still widely cultivated and sometimes it collected. So these both application can be very helpful to differentiate of two closely related species of Tinospora. So these are two prominent examples of our in-house studies where we have studied the botanicals and we have used HPTLC in from quality control, adulteration to the different studies of botanicals. So along with that, we have worked on different botanicals and we have optimized the methods from mobile phase, visualization, as well as derivatization for the different chemical classes. And these methods can be a database for us in the future guidance also, where these methods further can be optimized for different matrices. So these are some of the methods. Along with that, we have experimented and we have provided the combinational approach when it comes to HPTLC technique. So it could be HPTLC-UV plus HPTLC-MS, where you have confirmed the uh, analytical fingerprinting with the help of MS characterization, scan or SRM technique, or it could be your HPTLC-DS plus UFLC-UV, where you have standardized and quantified the bioactive analytical marker, or HPTLC-UV plus DS plus MS, where you have characterized the different multi-class compounds in the botanical matrices. So this could be a very interesting approach when it comes to botanical and botanical supplements. So in conclusion, when it comes to botanical, these botanicals are having different challenges. But with the help of HPTLC, diversification of HPTLC techniques, along with a robust scientific investigation, we could solve different challenges which currently we are facing into botanical and botanical dietary supplement investigation. Which one is the very much authentication in supply chain? You can have authenticated supply of the plant or the plant material or the plant part, which is very essential. Along with that, optimization of your geographical sourcing or the seasonal variation reduction is very important, which can be very cost effective and very crucial when it comes to commercial R&D. Chemical assessment, definitely quality control of chemical composition. So a variation of chemical composition can be checked and also standardized. Also, batch to batch testing is one of the very beneficial technique when it comes to HPTLC, and it could be easily monitored along with the stability of your botanicals or your extract or your products or formulation. HPTLC-MS has been definitely a very choice of technique, and it has provided a very conformational tool for safety and efficacy when it comes to adulteration. It could easily identify the adulterated or the correct sourcing of botanical. Along with the characterization data are very essential and which could be easily provided with the help of a prominent or a robust development of HPTLC method and scrapping of the band and identification of band and compliance of regulation. The unadulterated and authenticated sources definitely will lead to a very robust and prominent development of the new product or the existing product, which can be very beneficiary for the botanical dietary supplement. So this way I'm ending my presentation by thanking my wonderful team along with our inspiration, Dr. Lal Ingorani, and a very, very special thanks to my colleague, Ms. Chetna. Without her, this work would have been not possible, along with Ganesh and Ajas. And this is my wonderful team who has worked with me on this all research. Thanks to Farmanza Herbal Private Limited for this opportunity and all research work we could do with this. This work would have been never possible without this great pool of talented scientists and their guidance, their collaborations. So I would like to thank to all of our collaborators for this HPTLC studies. A very special thanks to our valuable partner, Bhartiya Sciences. Thank you very much.